Hello and welcome to Biology Bites. Today we'll be covering proteins from the Biological Molecules chapter with Laura. Hi Laura.
Hi.
So what are proteins?
Well, proteins are substances whose molecules are made of many amino acids linked together in long chains.
Amino acids?
Yes, every amino acid has a central carbon atom attached to 4 different chemical groups. An amino group, NH2, a carboxyl group, COOH, a hydrogen atom, and an R group.
What's an R group?
Well, the R group is a different molecule in different amino acids. It could be a whole range of different groups.
So how do amino acids join together?
It happens through a condensation reaction, where a hydrogen atom from one amino acid and one oxygen atom and one hydrogen atom from another amino acid join together to form a water molecule. The bond formed is called a peptide bond.
What if you wanted to break them up?
That would be a hydrolysis reaction, where the peptide bond is broken using a molecule of water. A hydrolysis reaction is the opposite of a condensation reaction.
What about dipeptides and polypeptides?
A dipeptide is a molecule made up of 2 amino acids joined together by a single peptide bond. A polypeptide is basically where more than 2 amino acids are joined together by peptide bonds.
Well, that's simple enough. What about the structures of proteins? Primary, secondary?
Tertiary and quaternary?
Yeah.
The primary structure is the sequence of amino acids in a polypeptide chain. The secondary structure comes in 2 common structures.
The alpha helix and the beta pleated sheet.
Exactly. Alpha helix has a coiled structure, and the beta pleated sheet is where different polypeptide chains become linked in parallel flat sheets. They're both held together by hydrogen bonds.
What's a hydrogen bond again?
A hydrogen bond is a chemical bond formed between the slightly positive charge on a hydrogen atom and the slightly negative charge on another atom of another molecule. An example would be the hydrogen bond between the hydrogen atom of one water molecule and the oxygen atom of another water molecule.
What about the tertiary structure?
Well, now the chain can be coiled and folded further, giving a complex 3D structure. The tertiary structure is held together by 4 possible types of bonds: disulfide bonds, ionic bonds, hydrogen bonds, and hydrophobic interactions.
And finally, the quaternary structure.
If a protein is made up of several polypeptide chains, the way that they are arranged is called the quaternary structure. It is held by the same kinds of bonds as the tertiary structure.
Well, thanks for that, Laura. We hope you found this helpful. There's more podcasts on this chapter on the blog, so stay tuned.